Every biologist I know has a crush, a secret crush on this molecule, DNA. And I'm going to tell you why. This is one, for me, it is a very important molecule from the class of biological molecules that are out there and that make life possible on Earth. This is clearly one of the most important ones, and I want to dedicate a tutorial on this one. And DNA is clearly, like I mentioned on the tutorial, on the nucleic acid tutorial, it is a nucleic acid, and specifically DNA carries the genetic information. It is the genetic material. So everything from your eye color, your height, to anything that you can inherit from your parents or grandparents. I'm talking about physical traits. It definitely comes through DNA. DNA is the code for all of that, all of that information. DNA is deoxyribonucleic acid. That's the abbreviation. And you have we talked about it in the nucleic acid tutorial, and I explained you why it is called deoxyribonucleic acid. But if I show you the monomer here, you probably remember what I'm drawing right here. This is a very important monomer in nucleic acids, and this is a nucleotide. Clearly, the nucleotide here in DNA is different from the one you find in RNA. Because of this pentose here, this center here, this sugar, in DNA is 2-deoxyribose. And why is 2-deoxyribose? Because here you have one less hydroxyl group. You only have a hydrogen, so one less oxygen, and it is in carbon number 2. Okay, that's why you have 2-deoxyribose. Well, before 1953, you have several scientists telling you that there is genetic information, there is genetic material being transmitted or inherited from parent to offspring, but none of those can tell you, well, there is actually a system of storing and transferring this genetic information. There is a molecule behind it, and this is how it works. They cannot clearly tell you and explain how it is done until 1953, where these two great scientists come with this clear model that opened a lot of doors to biology or biologists and scientists to figure out how we inherit blue eye color or height or anything from our family. So James Watson and Francis Crick proposed the structure of the DNA molecule that we know today. Now it is easy for us. We just open the book and we have everything, all the explanations there, but they did not know by then how this molecule was shaped and formed. And the model that they proposed, or the model for the DNA molecule, was that it consisted, don't forget the tutorial on nucleic acids, because I explained there the monomers for DNA is a nucleotide. So the model explains that the DNA molecule consists of 2 polynucleotide strands, and 2 polynucleotide strands mean several nucleotides, and 2 strands coiled around each other in a helical twisted ladder. That's the key thing here. You have probably seen a ladder, even climbed one. If not, I suggest you Google it. But the model of the DNA that we know today, and that was suggested at this point of time, was that you are looking at a ladder that twists around itself. That's the shape of DNA. That's the shape of the DNA we know today. So we know from now that the genetic information that was known previously is being stored like in this fashion. And I'm drawing here the shape of a ladder. So we have the 2 sides, and these 2 sides are comprised of the sugars and phosphates. And the middle, the steps of the ladder are the bases. They meet in the middle. They are bound to the pentose, okay? And they meet in the middle of the ladder. And I'm going to show you here on the picture on the left. the picture I have here, and I'm going to use the same color so you can understand where is my ladder here, because here you have the structure of the DNA twisted around itself. So here is my side of the ladder, my pentose and phosphates, and they are twisted around the bases here that I'm going to see two bases meeting in the middle. They are bound to the pentose and they meet each other in the middle. I want to use this time now to explain several characteristics of the DNA molecule that are quite important to know. And not only they're described in the Watson and Crick model, but they're also important to know for biology and molecular cell biology and classes that you see in medical school. So these characteristics, the first one is complementary base pairing. And the name says it all. I'm going to break it down for you. Base pairing. So as I explained in that ladder shape, okay, that we saw previously, the bases meet in the middle, okay? So you have one base on each side of the ladder, okay? On the two strands, you have one base here and another one here, like you see here. I'm going to put the red color here, or keep the magenta so you can see here, okay? These are my two bases, and they meet in the middle. They bind to each other with hydrogen bonds. Okay, very important. So the bases bind with hydrogen bonds to each other in the middle of the ladder, the DNA ladder, let's call it. And by complementary, I mean that they were only bind to certain complementary bases. Well, and this is very, very complicated word to say that, for example, cytosine will only bind to guanine. Okay, like we see here, cytosine and guanine base. And thymine will only bind to adenine. That's complementary. They're only complemented to each other. You will not see a cytosine binding with a DNA, adenine. Forget about that. And another thing that you are going to see is that when cytosine binds to guanine, you are going to find 3 hydrogen bonds. So here in the middle, if you see one cytosine on one side and the guanine on the other side, they're going to be bound to each other with 3 hydrogen bonds, okay? And when you see a thymine with an adenine, you are going to find only 2 hydrogen bonds, okay? Keep that in mind. In RNA, though, as I mentioned in nucleic acid tutorial, RNA, you do not find thymine. Instead, you find uracil. And the same applies to the RNA structure. When 2 bases meet, it's not the same shape, and we're going to see it, but the bases do meet and they do pair with each other through hydrogen bonds. But when they meet in RNA, instead of a thymine binding to an adenine, you see a uracil. And it's the same thing, 3 hydrogen bonds. While still on the topic of complementary base pairing, I want to talk about Chargaff's rule. And this applies to DNA because we talked about the double-stranded ladder shape form where you have an adenine bound to a thymine and a cytosine bound to a guanine. Okay, this is how you find things on this ladder, as you can see here on this picture on the left. So you always see, when you find one, you see the other bound on the other side, okay? And with this, he says that the number of purines in a DNA molecule equals to the number of pyrimidines. This to say, for example, if you have, this is a very simplistic way, but if you have 2 adenine bases in a DNA molecule, you are definitely going to find 2 thymines, okay, because they are complementary paired, you know, and if you found two cytosines, or in this case, this side, guanines, you will definitely find 2 cytosines on the DNA molecule. This is what I mean by the Chargaff's rule, and it's important to know for molecular cell biology. Another thing I want to still mention on this topic is that in some cases you have heard about self-complementary base pairing. And this is what we see in RNA, because RNA usually is found in single strands, not in double strands like this ladder. You see one strand here and another strand here. In RNA, usually it's found in a single-stranded form. And sometimes, because you have the bases on one side, for example, you have the bases on this side, when the molecule actually bends, for example, I'm going to try a draw here. For example, if this single-stranded molecule tries to bend this way, the bases are going to meet here in the middle, and they are going to bind to each other the same way you see here. An adenine with, in this case, uracil, if we're talking about RNA, and a cytosine with a guanine, and they're going to bind the same way. This is what we mean by self-complementary base pairing. And these loops, we call it loops, are found usually in RNA. So now we're going to talk about the new characteristic of DNA that we find in DNA molecules, and this is antiparallel orientation. This is what we call, and the name says it all. Again, in sciences, you always look at the name, and it usually says it all. So antiparallel orientation means that you are going to find the phosphodiester bonds that I just explained in nucleic acids tutorial, the ones you find here between the pentoses, or the nucleotides that bind. These are the bonds that are binding the nucleotides this side of the ladder. If we look at the DNA molecule, this side of the ladder, these bonds here, the phosphodiester bonds, are keeping it stable, are binding the nucleotides together. And then in the middle of the ladder, you have the hydrogen bonds between the two bases. Okay, so you have two types of bonds, the hydrogen bonds and the phosphodiester bonds. But looking at the sides of the ladders, you are going to be, I'm going to write down hydrogen bonds. You know already, but just to have a clearer idea. So on the phosphodiester bonds here, you're going to see that they have different orientations. So you're going to find a 3' to 5' phosphodiester bonds. And then you find, this is why you have antiparallel, it's 5' to 3' phosphodiester bonds. And what do I mean by this? Well, on the nucleic acid tutorial, I taught you how to count the carbons when you do the phosphodiester bonds, or when you look at the phosphodiester bonds. So if you start counting on the first position, or the 1' position, you always have a base attached. So I have my thymines here attached to my first one, and start counting 1, 2, 3, 4, and my 5th one is here. Same thing here, 1, 2, 3, 4, and my 5th one is here. Okay? This is what I mean with, okay, at this end, or this terminus, this end of this ladder, the side of this ladder here, the end of this molecule, I find a 3' or carbon in the 3rd position. So I call it 3' end. Okay? And here, at the end of it, I find a carbon in the 5th position. So I call it 5' end. That's why you have the 3', 5' end. 3', 5' phosphodiester bonds going in this direction. Okay? This is why you have two different ones. And then you start counting the same thing here. First position, 1, 2, 3, 4, and the fifth carbon here. And again, 1, 2, 3, 4, fifth carbon here. And when you look at this terminus, you find a fifth position, a carbon in the fifth position. So 5' end. This is a 5' end of the molecule. And here you find at the end of this terminus, you find a 3' end. As you can see, if you look at these two parallel sides of the ladder, of my DNA ladder, you can see that on this side here, you have a 3' end, and on this one, a 5' end. And as you go, they are antiparallel. So different orientations, you are going into different orientations. That's why you have antiparallel orientation. This side here is clearly a 5' to 3' phosphodiester bond. Okay, look at the nucleic acids tutorial because you will understand the phosphodiester bond. I explained it a little bit more clearly there. Okay, and if you look at the molecule here, this is that ladder with more detail. So you have two sides here. I'm going to use this color. So this is the other side of the ladder. Okay, and it is happening the same thing. So you have 3' end and 5' end here, and oriented in an anti-parallel fashion, you have 5' end and here a 3' end. And in the middle, you can clearly see the bases being bound to each other with hydrogen bonds, okay, like I explained previously. Now quickly going through these two very simple processes, even though they have complex names, denaturation and renaturation, these are also characteristics of this molecule or DNA. And if we look again at the structure of the ladder structure, what's going to happen when you have denaturation of this molecule is that you are going to disrupt the the H bonds, so the hydrogen bonds. This is when you destroy these, or you break these bonds between the bases. So the bonds, the H bonds between the bases. A lot of BBBs in this sentence. Anyway, what I mean with this is that you are going to go from one double-stranded DNA molecule, two strands, one on this side, as you can see, and another one on this side. This is what I mean by double-stranded. So you're going to go from one double-stranded DNA molecule to two single-stranded DNA molecules. So you're going to end up with two single-stranded DNA molecules. This is denaturation, very simple process. And the conditions in the environment that you need in order to cause denaturation of DNA molecules is heating. When you heat the environment, when you increase the temperature in the environment, we're going to talk about this a little bit later on. When you actually increase the temperature, you can break these bonds. You can disrupt the H bonds, the hydrogen bonds in DNA structure, in the DNA structure. Another condition that in the environment that can definitely disrupt is enzymes. Not a condition, but these molecules, these structures are able to disrupt also those H bonds, and some example is helicases. They are present in transcription and they unwind the parental DNA structure. You're going to see that later as well, I promise you, on tutorials if you're still interested, of course. But another condition of the environment is basic pH. If you increase the pH in a solution, you will definitely see that DNA, or the double-stranded DNA molecule will become 2 single-stranded DNA molecules. Another molecule that can cause denaturation is urea. Okay? Well, renaturation means the opposite. When you go from 2 single-stranded DNA molecules back to 1 double-stranded DNA molecule. And of course, if these conditions go to the opposite, back to normal conditions of the environment, of course, these two single-stranded DNA molecules are going to be able to restore the hydrogen bonds and become again a 1 double-stranded DNA molecule. Well, hyperchromicity, another scary word. Do not be scared. It's very simple, and I'm going to try to explain to you. It's actually when you increase the absorbance. Oh, sorry, let me correct here. The absorbance of light in a molecule due to some sort of change. In case of DNA, hyperchromicity happens when you denature the molecule. So during denaturation, like I explained previously, denaturation. And in denaturation, like I explained, means that you are going from a 1 double-stranded DNA molecule to 2 single-stranded DNA molecule due to the breaking or disruption of the hydrogen bonds. So here you have hydrogen bonds. And because of that, you also are bringing these 2 strands closer together. And therefore, you have here a denser or a more dense DNA molecule or more dense solution. In this case here, because you do not have the hydrogen bonds bringing these 2 strands together, they're separated. Let's say in this case, I increase the temperature, denature the molecules, they're separated now. So I'm decreasing the density. So this is dense, less dense. And when I do this, I'm actually, let's review a little bit of chemistry very quickly. Density equals mass over volume. Okay, so when I have an increase in density, and I still have the same mass in the solution because I'm not altering the mass, I only have to alter my volume here or change my volume. And in this case, what's going to happen is that I am going to have to, in order to have an increase in density, I am going to have to decrease my volume. And in order to decrease my density, I have to increase my volume. So to decrease density, I increase my volume. Okay, just looking at, you simply look at the formula, very basic. So if you are increasing your density, you're clearly not touching the mass because the mass of the DNA molecule stays the same in the solution, for example. But you have to alter your volume in order this is going to change the volumes. So the volume here, in order to have an increase in density, has to be going down. That way, when you divide by the mass, it's going to be a higher value than here where the density is decreased and the volume is higher. This to say, all of this to say that the light absorbance here is lower, so lower light absorbance, okay, and here it will increase, so higher light absorbance. So this is what I mean by hyperchromicity of the DNA. When you denature the molecule, you will actually increase the light absorbance means that if you have more molecules, more DNA molecules, more volume, you actually have more molecules absorbing the light. This is what it means. It is time to look at this graph. Do not be afraid. It's very simple graph, but it basically relates the two concepts that we just discussed: hyperchromicity and denaturation. And if you look at the left side of the graph, you see that this is the increase in ultraviolet absorption, and we're dealing with it in percentage. So you go from 0% to 100. And it is important to make this point here that DNA molecules, when you are studying absorbance, you study at a 260 nanometers wavelength. Okay, this is the wavelength of the light we're using here. Okay, and it's ultraviolet. Another thing to look at is the temperature here that we're working with it in degrees Celsius. And as you see, we will be increasing, going from here to here, increasing the temperature and seeing the effect that it has in absorbance. So we're relating absorbance to temperature and vice versa. And looking at the concepts that I just explained previously, looking at my double-stranded DNA molecule, when I increase the temperature, going down the graph, I'm increasing the temperature, I am producing this. So I am producing single-stranded DNA molecules that are happening due to the disruption of the hydrogen bonds. Okay? So keep that in mind. And as I also mentioned previously, that double-stranded DNA molecules are able to absorb less, so they absorb less light than single-stranded. Okay, single-stranded DNA molecules. Okay, so knowing that, if you look at this graph, as you increase the temperature, you are denaturing. So this is denaturing. You are denaturing the double-stranded DNA molecule. And as you denature it, you are increasing the absorption, right? You are increasing the percentage of absorption of light, of light that these molecules are doing, because you are going to this form here, okay? So if you look at the 50% here, this is 50% of absorption, meaning that 50% of the molecules that are being studied are actually absorbing 50% of the light, meaning that at this stage of the game, half of the molecules are in this form here, so half are double-stranded DNA molecules and half are single-stranded DNA molecules. This is what 50% here means. And you can also say that here at 50% of absorbance, if you go down and look for the temperature, you have 85, but more detail is 84.9 degrees Celsius. That's what we called melting temperature of the DNA molecule. And this is where you find, if you want to define melting temperature, this is where you say that that's the point of the game where you find half of double-stranded DNA molecules and half are actually in the form of single-stranded DNA molecules. Half of the molecules are already denatured. This is what it means. And if you go up, so you are increasing more and more the temperature, you are seeing that you are reaching 100% absorbance, meaning that all these molecules are becoming, all the double-stranded DNA molecules are becoming single-stranded at this point here, okay? So at this point, you have 100% absorbance, and because 100% of the molecules in the solution are found in the single-stranded form. I want to still use this curve to explain something about hyperchromicity and denaturation of the DNA molecule. And what I want to do with this is draw another curve here. And this curve is going to represent the another DNA molecule. And what I want to do here is say that this is my A DNA molecule, and this is the curve for my B DNA molecule. And if I look at my A DNA molecule, I can tell you that my melting temperature, or the melting temperature for the DNA molecule A, is going to be 84.9, okay, degrees Celsius. If I look at the B DNA molecule and I go meet at a point of 50% of absorption, where I have half of my molecules as double-stranded and half of my molecules as single-stranded, okay? And I go all the way down, meet at the curve, go down to my temperature and read the value, the temperature value. And I could say here that my melting temperature for my B molecule is going to say 88 degrees Celsius. Okay, so when I say this, I can tell you that the temperature needed to reach the melting temperature of the DNA molecule B is higher than for A, meaning that this molecule here is, I'm going to use another color because it's hard to see. This molecule here is heat resistant. So my B molecule is heat resistant or temperature resistant. You can also say that. And what I mean with this is that I need more temperature for my B molecule in order to break these bonds and have half as double-stranded and half as single-stranded. So I need more temperature for the B molecule. And why this happens, so there is something that we look at in this case that is the ratio of the guanine and cytosine and the adenine and thymine bonds. If I have more guanine and cytosine bonds in the middle of my ladder, I'm going to have more hydrogen bonds, and therefore I need more temperature to break those bonds. That's why my B molecule, I can say that my B molecule is going to have more of the guanine and cytosine bonds than my A molecule, for example, my DNA A molecule. Okay, so what I can also write, knowing this, is that the ratio, let me write, so this is not right. So the ratio of guanine and cytosine and adenine, thymine bonds in my B DNA molecule is higher. So higher, I have more guanine and cytosine in my B molecule than the ratio for the same bonds in my A molecule. My A molecule definitely has less of the guanine and cytosine bonds. Therefore, it's easier to break. So I need less temperature to break my molecule, or to bring it to a single-stranded form, okay, to break those hydrogen bonds. This is what I wanted to explain using this curve as well. I want to use these two illustrations to tell you that there are different forms of DNA you can find out there. It's not just exclusively the helical shape one that I just mentioned. This ladder structure that I explained before, the side chains can have different forms depending on the environment. And this is what we're looking at right now. And for BDNA, for example, this is the, let me do a better ladder here. Yeah, it looks better like this, a little bit cleaner. My BDNA, actually, this is the structure that you see here on the left. As you can see, this is the helical structure where this ladder will twist around itself. So it looks like kind of like this. I'm not a good drawer, or artist, but you can see here I'm highlighting in red. This is the side chains, or the pentoses and the phosphate groups of my DNA, and they're assuming this shape here. And this is a BDNA. And you can find this type of DNA in aqueous solutions, and as the name indicates, or aqueous environments, environments and solutions as well, but environments. And aqueous, by aqueous, I mean environments that have H2O or water. Hopefully you know that formula. But anyway, another thing that it is important to mention about DNA is that this is the majority, as you probably are thinking by now, majority of DNA in a cell. So the majority of the DNA that you find in a cell actually looks like this one here, like bDNA. So this is my bDNA. Another form you find DNA in cells as well is zDNA. And the side chains here, the sides of the ladders here in zDNA actually form shapes looking like kind of crooked, looking like a Z shape. As you can see here, I'm highlighting in yellow. This is my zDNA. And this form of the molecule is actually DNA, or is found in DNA bound to certain proteins. And we're going to see that later in other tutorials, certain proteins here, okay? So just for you to have an idea that DNA is found in different shapes and form, not only the helical form, the beautiful helix that you're used to seeing, you actually have other forms, and this is what I wanted to show you here. Important thing to mention is that DNA is found in circular and linear forms. So you can find in the circular form, sorry, my English, I think it's the time of the day or the night that, you know, I need coffee basically. So the circular DNA means that it's going to be found, the molecule is going to be found in a circular shape. Nothing else to say. It's a very simple model. And you find that on viruses. And on viruses, you usually find single-stranded molecules, okay? Keep in mind the single-stranded that I explained previously. So only one side of the pentoses and the phosphates with some bases and no ladder shape like we see in eukaryotic form of DNA. Prokaryotes have also circular DNA, the one found in the nucleoli, like I mentioned in, you know, the prokaryotes tutorial, but in this case you can actually find it in the ladder form. So it's circular shape, but it's in the ladder form, not like in viruses where it's only single-stranded. So ladder form, double-stranded. You can also find circular DNA in eukaryotic cells, and like I also mentioned in other tutorials, other lectures, that you find this type of DNA, the circular shape or the circular form, in mitochondria, for example. The DNA there is actually circular shape. That's how you can relate it to prokaryotes, mitochondria to prokaryotes, because it is also circular DNA. And some other plastids, like chloroplasts in plant cells. You can also see that the DNA there is in circular shape or circular form. Another thing to say is linear DNA. So when I say linear DNA, is actually the molecule is on a line form. There is no circle. It's a line form. And you see this in eukaryotic cells. The DNA found in the nucleus is actually in the linear form. And it's two single-stranded DNA molecules joining forces, or joining or being bound together by hydrogen bonds, like I mentioned way before, as you can see here, bound by hydrogen and forming the double-stranded ladder that we know very well, but all in the linear form. Another thing that I would like to mention is that this linear DNA found in the nucleus are then found in chromosomes. So one chromosome is actually one long, if you stretch it, because the chromosome, imagine like a line, if you get a line or a cord and you compact that into the form of a chromosome. That's how you find the DNA in eukaryotic cells. And it's more or less 2 meters of linear DNA. And when they compact, they form the we're going to talk about that later, the chromosomes we know. Another thing to finish this tutorial that I want to use and talk very briefly about the DNA superhelix, but because it's important to understand that the superhelix of this molecule means that the molecular structure will form another helix. The helix that you know, the very simple helix that I explained, will actually form another helix around that helix. That's why we call it superhelix. And it's a very useful process and structure because, and I'm going to explain you why. But for now, keep that in mind because we will need this for another topic that I'm going to mention later on in different tutorials, especially when we talk about how the DNA is packed in eukaryotic cells. And I'm going to bring this up again. But in any case, just keep in mind that the DNA structure is a superhelix, is a helix around, forming a helix around the helix that we know. But I'm sorry about the drawings, but I think it gives you an idea what I'm talking about. If not, please let me know. I'll try to do a better one next time. But the usefulness of this superhelix is that it's compacting DNA. So it's actually packing the DNA within the cells, all those cells. Because as I mentioned, if this linear molecule has 2 meters of length, how am I going to pack in a very, very small cell that is only like several micrometers long? So I have to pack it. So it's very useful to have this superhelix structure. So that's why it reduces space taken by the DNA in the cell. Another thing that I would like to mention is that topoisomerases are these enzymes that are going to uncoil the DNA or underwind or overwind, whatever you want to call it, during replication or transcription. The process is when you want to do another copy of DNA. So you have to uncoil this really packed molecule in order to be able to replicate it. And this is where topoisomerases come in. And we're going to talk about these enzymes in more detail later on.